Hello and welcome to today's lecture on the histology of the intestinal epithelium. In this detailed script, we will explore the various aspects of the structure, function, and regulation of the intestinal epithelium. So let's dive in. The intestinal epithelium is a single layer of cells that line the innermost surface of the intestinal wall. It forms folds, known as villi, which increase the surface area for nutrient absorption. Each villus is covered by specialized cells called enterocytes, which have microvilli on their apical surface that further enhance absorption. Below the enterocytes is the lamina propria, a connective tissue layer that houses blood and lymphatic vessels, immune cells, and nerve endings. The intestinal epithelium is continuously self-renewing, thanks to the presence of intestinal stem cells. These stem cells reside at the base of small invaginations called crypts, which are scattered along the epithelial layer. They possess the ability to divide and give rise to both new stem cells and daughter cells that undergo differentiation. This process ensures the constant replenishment of the intestinal epithelium. The proliferation of intestinal stem cells is tightly regulated to maintain tissue homeostasis. WNT signaling is a crucial pathway involved in promoting stem cell proliferation. It acts through the activation of beta-catenin, which enters the nucleus and regulates the transcription of specific target genes. Additionally, growth factors secreted by neighboring cells, such as the epidermal growth factor, EGF, also play a role in stimulating stem cell division. As daughter cells move up from the stem cell zone towards the villi, they undergo a series of differentiation steps. One main pathway involves the differentiation of these cells into specialized absorptive enterocytes. Other cell types, such as goblet cells secreting mucus, enteroendocrine cells producing hormones, and Paneth cells responsible for antimicrobial peptide secretion, also arise from the stem cell lineage. This differentiation process is tightly regulated to maintain the balance of different cell types within the epithelium. WNT signaling plays a crucial role not only in stem cell proliferation but also in maintaining the intestinal epithelium. Loss of WNT signaling leads to a depletion of stem cells and a disrupted balance of cell types, resulting in impaired epithelial renewal and digestive function. The WNT pathway interacts with various other signaling pathways to coordinate stem cell dynamics and tissue repair. Another important signaling pathway involved in intestinal epithelial homeostasis is the Notch pathway. Notch signaling regulates cell fate decisions by promoting the differentiation of secretory cells such as goblet cells and enteroendocrine cells while inhibiting the enterocyte lineage. Disruptions in Notch signaling can lead to altered cell differentiation and the development of intestinal disorders. In certain pathological conditions, such as tissue repair or cancer progression, epithelial cells can undergo an epithelial mesenchymal transition, EMT. During EMT, cells lose their epithelial characteristics and acquire mesenchymal properties, allowing them to migrate and invade nearby tissues. This process plays a crucial role in wound healing but can also contribute to cancer metastasis when deregulated. Intestinal crypts and villi are distinct structures within the intestinal epithelium. Crypts contain stem cells at their base and are responsible for epithelial regeneration. Villi, on the other hand, are finger-like projections that increase the absorptive surface area. They contain a central core of blood vessels and lacteals, specialized lymphatic vessels that transport dietary fats. The microenvironment surrounding intestinal epithelial cells, also known as the niche, plays a crucial role in regulating their behavior. Factors such as extracellular matrix components, immune cells, and growth factors influence cell proliferation, differentiation, and migration. Crosstalk between the epithelial cells and the microenvironment ensures proper tissue function and repair. In addition to cell division and differentiation, apoptosis and cell shedding also contribute to the turnover of intestinal epithelial cells. As cells reach the tip of the villi, they undergo programmed cell death and are subsequently shed into the intestinal lumen. This shedding process is tightly regulated to maintain the appropriate number of cells within the epithelium. The stem cell niche is a specialized microenvironment that supports and maintains the function of intestinal stem cells. It provides physical support, soluble factors, and cell-cell interactions that regulate stem cell behavior. Components of the niche include mesenchymal cells, immune cells, extracellular matrix proteins, and growth factors. The intestinal epithelium is constantly exposed to a vast array of microorganisms and potentially harmful substances. Immune cells, such as lymphocytes and macrophages, play a vital role in defending against infections and maintaining intestinal homeostasis. They help coordinate immune responses, regulate inflammation, and contribute to the repair of damaged epithelial tissue. In conclusion, the intestinal epithelium is a dynamic and complex tissue that ensures nutrient absorption, protects against pathogens, and maintains tissue homeostasis. Understanding the intricate structure, regulation, and cellular dynamics within the intestinal epithelium is paramount for diagnosing and treating various gastrointestinal disorders. Thank you for joining me in this lecture, and I hope it has provided you with valuable insights into the histology of the intestinal epithelium.